Welcome back, everyone, and I'm pleased to introduce our next speaker, Dr. Amadou Gaye from the National Institutes of Health. Dr. Gaye completed his undergraduate studies at the Mannheim University of Applied Sciences in Mannheim, Germany. He then moved to the United Kingdom, where he obtained a Master of Science in Bioinformatics at the Department of Biochemistry, University of Leicester, and a PhD in genetic epidemiology under the supervision of Professor Paul R. Burton and Professor Martin D. Tobin at the Department of Health Sciences of the University of Leicester. After moving to the University of Bristol, he completed postdoctoral research in genetic epidemiology and data science at the School of Social and Community Medicine. In 2014, Dr. Gaye moved to the National Institutes of Health in the US. He is currently a staff scientist in the lab of Dr. Gary H. Gibbons, which is an interdisciplinary group that investigates the effects of genetic, environmental, and social determinants of health on cardiovascular disease mortality and morbidity. Please join me in welcoming Dr. Gaye.
Hi, good morning, everyone. My name is Amadou Gaye. I work at the NIH, the National Human Genome Research Institute of the NIH, and my work is primarily on cardiovascular disease. I want to thank the organizers of this conference for giving me the opportunity to share with you some of the work that we're doing in our group. The work that I'm going to present to you today, it's more in the sphere of nutrient genomics. We're trying to find a signature of sodium intake and link that transcriptome signature to cardiovascular traits. This is the outline of my talk. I will go through the background, tell you what is the motivation of this study and the aims. Then I will present the data, describe the transcriptome data, the dietary data, and some other data descriptions. And then I will succinctly talk about the strategy and methods that we used. And finally, I will present the results of one of the methods, the random forest analysis. Then I will show you the weighted gene co-expression network analysis, which is just a network analysis to find network modules that are related to sodium intake and cardiovascular and the cardiovascular variables that we investigated. Then we'll go through the pathway enrichment analysis to show you molecular pathways that are enriched in the modules that we identified. Finally, we'll finish by giving you some biological insights, describing what we have found in the context of the biology, because all of these was the results come from bioinformatics and biostatistics, so it's important to link it back to biology to give it the real meaning. It is well established that increased sodium intake is associated with an increased propensity of blood pressure and the development of hypertension, as well as the promotion of end organ complications, such as chronic kidney disease. In this analysis, we look into kidney function and use eGFR as a proxy measure for kidney function. In the literature, there is an evidence to suggest that African Americans exhibit a higher risk of developing hypertension at an early age, so they have an early onset of hypertension, and they have also a greater propensity to develop hypertension-induced complications, such as chronic kidney disease. There is also a large body of evidence that indicates African Americans are more prone to salt-sensitive hypertension. Now, there are also socioeconomic factors and social determinants of health that affect access to a healthy diet. We all know how healthy diet is relevant overall for health and disease. And the population that we just mentioned, African American and usually also underserved populations, tend to live on food deserts. And they tend also to live in areas that have a higher density of fast food restaurants, which do not usually serve healthy food. And they also tend to live in areas that have a low number of supermarkets with affordable fresh fruits and vegetables. Now, all of these factors would predispose the person living in racially segregated areas to be limited to diets that are high in sodium and that are usually also calorie dense, meaning diets that is high in sucrose and fat and so on. Some studies have reported that African Americans consume diet high in salt, sugar, and fat above what is suggested in dietary recommendations. So the aim of this study, we know that although population studies have documented a link between sodium intake and blood pressure and hypertension, the molecular pathways that mediate that link are still not very clearly defined. So our aim is multifold. First, we would like to define a transcriptome profile associated with sodium intake. Then we want to evaluate the relationship between that transcriptome profile and other nutrients that are usually correlated with sodium intake, such as fat, sucrose, and calorie, because we just said in the previous slide that diets that are high in sodium intake tend to also be high in fat, sucrose, and calorie intake. So it makes sense then to investigate the relationship between the transcriptome profile that is linked to sodium intake to see if that transcriptome profile overlaps with fat, sucrose, and calorie intake. Then the third aim is to find out if the transcriptome profile that we found related to sodium intake is also linked to hypertension and blood pressure. Finally, we said that chronic kidney disease or kidney failure or decline in kidney function tend to be downstream of hypertension. So we want to link the transcriptome profile to eGFR, which, as I said, is a measure here of kidney function, to see if that transcriptome profile that is now linked to dietary variables and hypertension is also related to kidney function. To start with description of the data, I would like to tell you a bit about the transcriptome. The transcriptome data consists of messenger RNA sequencing of 582 whole blood samples. Those samples are from two studies. One of the studies, the gene forecast study, 103 samples are from the gene forecast study, which is a study of African Americans recruited in the Washington, D.C. area, aged between 35 approximately and 65. And that transcriptome data were sequenced on the Illumina HiSeq 2500 and the Illumina HiSeq 4000. And we sequenced it to a great depth because we would like this data to be amenable to novel findings. So shallow sequencing won't allow you to uncover a lot of novel signals. The second data set consists of 179 samples from the Minority Health Grid study, and these individuals were recruited at the Morehouse School of Medicine in Atlanta, Georgia. It's African Americans also between the age of 30 and 65. And these data were sequenced on the Illumina HiSeq 2000 at a depth similar to the one of gene forecast. Now, for both these data sets then, after sequencing, the messenger RNA expression was quantified using a pipeline developed by the Broad Institute. And that pipeline is actually used by one of the leading studies of gene expression, which is the GTEx consortium. Now, because I said that we have two data sets, and then that means there is a possibility of batch effect. Then the first step, then the most important step at the beginning, was to make sure that there is no batch effect, that what we are finding from our analysis is not a difference between the two data sets, but that resulting just from the way the samples were sequenced, because they were sequenced in different platforms. So here I'm showing you a PCA plot of the two data sets, the gene forecast in red and image grid in black, to show that after going through all the quality controls, the samples that we are left with do not show any sign of a batch effect, because actually the two sets of samples do overlap and are not separated. So the analysis then was conducted with protein coding messenger RNAs, protein coding genes. Throughout this presentation, I'm going to call messenger RNAs is genes in short, but what I meant to say is really messenger RNAs. So we use 16,667 protein coding genes. The dietary data that we use in this analysis was assessed for 472 participants, and this is a smaller subset of the individuals we have messenger RNA sequencing data for. These 472 participants, the dietary data was assessed with a short food frequency questionnaire that was developed from a long questionnaire, and it was previously designed for the USDA, the Delta Nutrition Intervention Research Initiative 7, known as Delta NIRI. We use the total salt dietary intake from the contributions of each of the 158 food items considered in that questionnaire. Now, one important point here is that the nutrient intake distribution in our data are similar to those reported by previous analysis of 436 samples from the Jackson Heart study that used the same food frequency questionnaire. This point is important to just say that the distribution that we see do not deviate from what is known usually from this population. This is a succinct data description of some of the variables that were the most relevant one for this analysis. We start with the demographics. The mean age was 47, and there's a standard deviation of 10. There were more females than males, and that is not surprising usually in many epidemiological studies or even clinical studies. Dietary intake variables, I give here the sodium intakes in milligram, fat intake in gram, sucrose, and calorie intakes in kilocalories. The cardiovascular traits that we consider are hypertension. It's a categorical variables where we just consider individuals as non-hypertensive or hypertensive. I'll come back to the definition of hypertensive and non-hypertensive. There were slightly more non-hypertensives or controls than cases. And we report also the systolic blood pressure, the diastolic blood pressure, and the EGFR. Majority of the individuals had a kidney function that was above the threshold that what is considered well-functioning kidney, which is above 60. It's usually, below 60 is usually considered as a decline in kidney function. So the mean here is 97, which means the majority of the individuals here had a kidney function that was great. Body mass index, most of the individuals included in this study were obese. The mean is 31 and the standard deviation is 7. Here is another way of viewing the data. I'm showing here the correlations between the variables. The dietary variables, sucrose, fat, and calorie intakes are positively correlated, as we said, and this is not surprising. As we said before, diet that is usually high in sodium intake tend to be also high in sucrose, fat, and calories. And the blood pressure variables, systolic and diastolic blood pressure, as you would expect, are very strongly correlated, and they correlated also with hypertension. EGFR is negatively or inversely correlated with hypertension, systolic blood pressure, and diastolic blood pressure. This is also what you would expect because, as we said before, as blood pressure rises, kidney function tends to decline. So that's what we see here. And there is a positive correlation between hypertension, blood pressure, and BMI, because we know also that as BMI increases, people tend to more fall towards hypertensive states, or at least have an increase in blood pressure. And you find also more age also increases. Propensity to hypertension increases with age, so you find also here a positive correlation between blood pressure and age. So I'm going through the strategy and the methods. This talk is not a methodological talk, so we're not going to talk in details about the approaches we used here. We're putting here this slide just to indicate at each step what statistical methods that we used. First, we wanted to link messenger RNAs and sodium to identify a set of genes or protein coding genes that can predict sodium intake or associated with sodium intake. In this case here, sodium intake, fat, sucrose, calorie, all of the variables that were continuous were categorized, and then we're looking into the tails here. We're interested in individuals with a high sodium intake versus the individuals at the lower tertile of sodium intake. So basically sodium, fat, sucrose, and calorie were all categorized into the top tertile versus the bottom tertile. So the first analysis here is then to use random forests to define a transcriptome profile that predict or classify top tertile versus bottom tertile of sodium. And in the second step, we said that once we have found a transcriptome profile that can predict sodium intake, we would like to know if that transcriptome profile is also related to fat, sucrose, and calories. There also we use random forests to investigate that relationship. Then a third step, and that was also the third aim, we want to evaluate the relationship between the transcriptome profile and 4 of the cardiovascular traits that we mentioned at the beginning, that is hypertension, which is a categorical, cases and controls. Forgot to mention that actually the cases are hypertensives are categorized as individuals with a blood pressure greater than 130 and 85, and they, or if they're on medication, they're also considered as hypertensives. And the controls are individuals with perfect blood pressure without medication. They must be 120 to 80 systolic diastolic blood pressure, and the kidney function must be also perfect, above 70. So we evaluate then the relationship between the transcriptome profile now linked to the dietary variables, its relationship with these cardiovascular variables. And finally, we run a network analysis. Now that we have a set of genes that we know are related to dietary intake and are related to cardiovascular traits, such as hypertension, systolic, and EGFR kidney function, we want to know if there is a network of co-expressed genes within that set of genes. So basically that is identifying clusters of co-expressed genes. And then finally, we do then the pathway enrichment analysis within those models. So now we're going through the results. The first step, as I said, was to link sodium intake to messenger RNA. So we ran that analysis and found a set of 948 genes that predict sodium intake, meaning that can classify upper tertile and bottom tertile of sodium intake with an area under the curve of 0.82. So we have now 816 genes that can predict sodium intakes. We test those 816 genes, how well that set of 816 genes that predict sodium intake can predict fat intake, sucrose intake, and calorie intake. And we run a random forest analysis with 1,000 trees. Exactly the same parameters that we used in the first model fitted to predict sodium. That's the same parameters that we use to test the relationship between these 816 genes and fat, sucrose, and intake, and calorie intake. We found those 816 genes do predict fat intake with an area under the curve of 0.75. They do well at predicting calorie intake, 0.77, and less well at predicting sucrose intake, because the area under the curve for that model was 0.62, which is not very far from tossing a coin, which is a 0.5. So the take-home message here is that we found a transcriptome, a set of 816 protein coding genes that can predict sodium intake, and they can predict fat intake, and they can also predict calorie intake, and only to a lesser extent sucrose. The third step of the third results were that we want to know now if those 816 genes link to sodium, fat, and sucrose intake are also related to hypertension, systolic diastolic blood pressure, and kidney function, EGFR. That is because we also mentioned earlier that there is a known link between poor diet, a diet that is high in salt, fat, and sugar is considered a poor diet. If there is a link, that link is already established, the link between poor diet, hypertension, and EGFR. So it makes sense then to subsequently evaluate the relationship between those 816 genes that are related to dietary variables to see if they're also predictive of hypertension, systolic blood pressure, diastolic pressure, and EGFR. So we fit again a random forest model with exactly the same parameters and say, oh, can we predict hypertension, systolic blood pressure, and EGFR with these 816 protein coding genes? As the results indicate here, it seems like those 816 genes are related to hypertension because they can predict cases versus controls with an area under the curve of 0.72. And if we look into the tertiles, top and bottom tertiles of systolic blood pressure, we could classify systolic blood pressure with an area under the curve of 0.71. Those 816 genes are doing less well with diastolic blood pressure. Still, the area under the curve is close to 0.7. And EGFR is also predicted with an area under the curve of 0.71. Now, this is a summary of all what we discussed so far. We found 816 protein coding genes that are related to sodium intake and are also related to variables often correlated with sodium intake, such as fat, sucrose, and calorie intake. That transcriptome profile, we found it also related to hypertension, blood pressure, and kidney function, EGFR. So the next step was to find out if within these 816 genes, we can identify clusters of co-expressed genes, meaning genes that have an expression that is correlated. We use weighted gene co-expression network analysis to basically uncover those deeper patterns. And those deeper patterns are clusters of co-expressed genes. Because what random forest is telling you is that these set of 816 genes are related to these variables, but there might be substructures within that 816 genes, and that's what we're trying to uncover here. And those clusters, we call them network models, because you have a whole network of 816 genes, and within that network, you have some clusters of genes that are more correlated than others. So the analysis we conducted follows the WGCNA methodology. As I said, this is not a methodological talk, so we're not going to go into the details on how weighted gene co-expression network analysis works, but basically we can summarize it in 3 steps. At the first step, what you do in this approach is you construct a network. You construct a network of genes using all of the 816 genes that were identified by random forest. Then in that network, each gene or its messenger RNA is a node, and closely related messenger RNA then forms a model. So it's a network, and within the network you can see patterns of genes that are correlated, and they tend to be closer to each other, and those are the clusters I was talking about. Now, once you identify those clusters or models, you use hierarchical clustering to identify those models. You go through the network and use hierarchical clustering to find those clusters that we call network models. Now you identify network models that is completely independent from the last step that we are talking about here. You have model, let's say A, B, C, they're usually named after colors. Let's say you find a model, as we're going to show next. You find a model. It's an aggregated expression of genes. That aggregated expression, you would like to know if it is associated or correlated with any of the variables that we talked about here, the nutrient variables and the cardiovascular variables. So this is the results of the WCNA. To go explain what this graph means, if we, for example, look at the bottom, you see turquoise, that's the turquoise model. Here, what we found after the analysis, we found 4 network models. And those 4 networks models are the ones represented here by blue, yellow, brown, and turquoise. Each of these models, as I said, is an aggregated expression. For example, the turquoise model consists of 64 co-expressed messenger RNA, or just 64 co-expressed genes. Now, that turquoise model consists of the aggregated expression of those 64 genes that constitute one vector. If you can think about it in terms of principal component analysis, it is like the principal component that explains most of the variability or variance in the data. So that turquoise model, then you look whether that turquoise model is correlated with sodium, sucrose, as we're going through the x-axis here, calories, hypertension, systolic blood pressure, and so on. For each of these values, the top value here, for example, between turquoise model and sodium, the top value 0.43 on the bottom left corner, the 0.43 is the correlation between that model and sodium, meaning between the aggregated expression of the 64 genes that are in that model and sodium. And the values between the brackets are, the first one is the p-value after adjusting for age and sex. And the second p-value is the p-value after adjusting for age, sex, and multiple testing. Because you find a lot of models and you're testing each of the models, you have to penalize for that multiple testing. So you have then, for each of square here, it tells you the correlation between the model, the top value, the raw p-value, raw-ish, because it's already adjusted for age and sex, and the final p-value, which is a p-value adjusted for age, sex, and multiple testing. So what we find here is that 3 of these 4 models, the yellow, the brown, and the turquoise, are associated with sodium intake, sucrose, fat, calorie intake. They're all positively associated with those nutrient variables. And then they're negatively associated with hypertension, systolic blood pressure. And they are positively associated with EGFR. Oh, I forgot to mention that actually we adjusted also for BMI, because it is known that people who are larger tend to have larger food portions, and that can then, if you don't take that into account, it can introduce some skewing of the data. So basically, the amount of sodium and salt and sucrose and fat in a portion would be larger for someone who is having a larger portion. And it is known actually in the literature that the amount or the portion of food is usually correlated with BMI. So we adjusted for BMI, age, and sex. To take it back, to just say that actually the first p-value that you see here is adjusted for BMI, age, and sex, and the second p-value is adjusted for BMI, age, sex, and multiple testing. And yes, I should have indicated that at the bottom here. This is how the turquoise network model looks like. On the y-axis, you have the 64 genes that are in this network model, and on the x-axis, you have the variables. It is clear to see that we have a pattern here. There is a larger group of these genes that is positively associated with the dietary intake. That's the red, and that group of genes is negatively associated with the blood pressure variables, that's in yellow. And here we show you again another of the 3 models. We said it was the turquoise, the brown, and the yellow model. This is also patterns that we see in the brown network model. A set of these genes is positively associated with the dietary variables and negatively associated with blood pressure. And this is the yellow network model, where pretty much all the genes are positively associated with the dietary variables except 2, the one at the bottom. We then now first we have identified a set of 816 genes that are linked to dietary intake, and those 816 genes are also linked to hypertension, blood pressure, and kidney function. Then within that a network of 816 genes, we found clusters of co-expressed genes. Now, in those clusters of co-expressed genes or network models, we run pathway enrichment analysis. And here we are showing the most important results in terms of the turquoise model, the brown model, and the yellow model. I highlighted here 3 of the pathways enriched in these models that we're going to talk more in details about in their biological insights. So what this table is then summarizing is telling you the pathways that are enriched in each of these models. For example, for the turquoise model, you have the hyposignaling pathways that is significantly enriched in this model, and the p-value, the adjusted p-value, is less than 0.05. And in that same turquoise model, you have also renal carcinoma, and indicating something going on with the kidney, with the gene RAP1A. In the brown model, we're going to talk about the pathway Gaye-like receptor, and we're going to mainly talk about the TRAF3 gene that has been already highlighted previously in animal models. So now we're going to finish this talk by giving you some of the biological insights. What can we tell about our results looking into animal models that has been published previously? We start first with the TRAF3 genes that we believe is a potential mediator for unhealthy dietary patterns. What I'm showing you here on the table on the top right is the correlation between the TRAF3 genes and sodium, sucrose, fat, calories, hypertension, systolic blood pressure, and EGFR. You can see that the expression of that gene is positively correlated with sodium, sucrose, fat, and so on, meaning the expression of that gene increases with increased intake of sodium, sucrose, fat, and calories. And it is negatively correlated with hypertension, systolic blood pressure, diastolic blood pressure, and positively correlated with EGFR, meaning the expression of that gene decreases with increasing blood pressure or increasing risk of hypertension. Now, that TRAF3 gene, TNF receptor associated factor 3 is the name. It's a hub gene in the Brown network models that we found within the set of 816 genes. Now, it's a hub gene. It means that it's a gene that is at the center of that model. A hub gene is a gene that has the most connections within a cluster of genes. So if you were to look at it graphically, you will say this gene must be important because it sits at the center of a network of genes, and it is connected to most of the genes in that model. So what is known about TRAF3? TRAF3 is known to be involved in the signaling of pro and anti-inflammatory biomarkers such as cytokines, toll-like receptors, and NF-kappa B. And two of the studies that I show here on the bottom right, one of them indicate that high salt consumption has been shown to recruit toll-like receptors and TRAF3 to endothelial cell lipid rafts. Now, that study indicated also that TRAF3 has pro-inflammatory activity in obese mice, and the activity seems to be controlled by a dietary and obesity-associated factor. So there's definitely a connection between that gene and the phenomenon that we are investigating here and the rest of that model. Further about that TRAF3 gene, through the activation of the RAS system, an abnormal activation of TRAF3 interacting protein 2, which is a TRAF3 IP2, may operate as a mediator of aldosterone and salt-induced cardiac hypertrophy and fibrosis. And actually, importantly, knockdown experiments of this gene, TRAF3 IP2, demonstrate that it actually inhibits VEGF-A production and vascular endothelial cell growth, which are all extremely important for blood pressure. An animal model also study showed that TRAF3 IP2 plays a causal role in vascular insulin resistance and dysfunction. So one of the take-home message is that, interestingly, TRAF3 is known to mediate the activation of TRAF3 IP2. So the gene that we found to be a hub gene, meaning an important gene in the models that we identified, is known to mediate the activation of the gene that we described here as related to aldosterone salt-induced cardiac hypertrophy, as reduction linked to the production of vascular endothelial cell growth, and so on. And it is also known that the activation of this gene TRAF3 IP2 by the gene that we identify results in the activation of NFB, AP1, as well as the overexpression of several pro-inflammatory, such as NF-kappa B and pro-fibrotic genes. Finally, on TRAF3, it has been demonstrated that the gene that it activates, the TRAF3 IP2, mediates interleukins, such as interleukin 17, signaling by binding to the interleukin 17 receptors, meaning IL-17R. Now, interestingly, although they were not in the network models, in the set of 816 genes, we have interleukin 17 receptor E and interleukin 17 receptor D. That means genes related to TRAF3 IP2 are within the set of genes that predict the sodium intake and are also related to blood pressure and kidney function. Some previous results actually revealed a strong association between fast food consumption and an increased number of Th17 cells in the circulation. And Th17 cells are actually characterized by the production of IL-17, which again link us back to that TRAF3 genes. It has been also reported that previously reported that a diet high in sodium increases IL-17 mediated inflammation, and that situation could worsen autoimmune diseases. Those are the papers that actually I put here on the right side that I'm talking about in red. So to summarize what we talked about here about the TRAF3 genes, we can say that in summary, TRAF3 seems to mediate unhealthy dietary patterns such as a high salt and high fat by promoting vascular dysfunction, that is impaired flow-mediated vasodilation, as a type of mechanotransduction. It promotes also pro-inflammatory cytokine and oxidative stress. It does that also by promoting metabolic dysfunction, such as obesity and metabolic syndrome. And then it's also promoting the predisposition to increased blood pressure and vascular remodeling, which is a hallmark of hypertension. And the second point that the second gene that we would like to highlight in our results is RAP1A, and the hyposignaling pathway that is enriched in one of the models, as we showed before, the Chilcot's model. The RAS-related protein RAP1A, which is the full name, is also a hub gene. And here on the right side, on the top right, I'm showing you the correlation between the expression of that gene and the variables that we consider. It increases with increased sodium, sucrose, fat, and calorie intake. It decreases with decreasing blood pressure and diastolic blood pressure, and it increases with EGFR. So, as I said, we explained already the concept of the hub gene. It means the gene that is potentially the most important in the network model by its number of connections it has to the neighboring genes. Now, RAP1 has been demonstrated to induce cytokine production in pro-inflammatory macrophages through TNF signaling, and it is highly expressed in human atherosclerosis lesions. And most importantly here, we have to say that RAP1A, which is the gene that we identified in the model, there is a knockdown of that gene, and when it is knocked down, it alters cytokine response and MAP kinase signaling and inflammatory responses in an in vivo analysis, in vivo experiment. Further on RAP1A and hyposignaling, it has been demonstrated that RAP1B is the major isoform required for VEGF-induced endothelial permeability and for the dynamic regulation of endothelial barrier. RAP1B knockout mouse developed cardiac hypertrophy and hypertension. That is a direct link, actually, because this knockout analysis then shows that if you suppress this gene totally, what are the consequences? What's the phenotype that you're observing? So the hypertension phenotype is here directly linked to this gene through that knockout experiment. And a study on rodent rats showed that hypoglycemia inhibits RAP1B expression and its activity, which leads to tubular cell injury in animals with diabetic nephropathy. Further on RAP1A and hyposignaling, the gene and that pathway, they appear to function in concert in mechanotransduction, establishing a channel for mechanosignaling from the extracellular matrix to the nucleus. And two of the take-home messages is that increased arterial stiffness results in an increase in systolic blood pressure, left ventricular workload, and hypertrophy. And these are some signs of chronic hypertension, because usually when someone has had elevated blood pressure for a long time, then they tend to have a dilated left ventricular or cardiac hypertrophy. And we have to say also that mechanoactivation of the hyposignaling pathway is induced by increased vascular stiffness. A number of studies from animal and cellular model systems make a strong case that the YAP-TAZ related to the hyposignaling activity can be modulated by metabolic and nutrient sensing pathways. So that means that diet is directly involved here because of that nutrient sensing pathways that then transmits the signal. A recent study demonstrated that an activation of the hypo pathway promotes hypertensive renal injury, again linking the kidney to the hypertension here. Interestingly, though, the TRAF3 and MAP kinase have been previously identified as upstream regulators of the hyposignaling. So here we're linking what we talked about in the TRAF3 gene to what we talked about with respect to the hyposignaling. That brings us to the conclusions. What we can say is that what we observe in our results is that dietary intake is likely to impact blood pressure and kidney function through several pathways. And those pathways actually involve multiple genes that act in concert in molecular pathways. Therefore, rather than investigating genes in isolation, That's why in this project, we use ensemble methods to identify clusters of genes involved in molecular pathways related to nutrient intake, hypertension, and EGFR. It would have been actually difficult to get to these findings by looking at the genes, each of them separately, assuming that they don't work in concert. Although that biomarker approach is very important and has yielded a lot of important results in many of the multifactorial conditions such as hypertension and kidney disease, it is important to comb larger and see what genes act in concert to lead to the phenotype that we observe. Finally, we identified key genes that are driving network models that are biologically plausible mediators of the connection between dietary intake, blood pressure, and EGFR. I would believe that our results provide a foundation for future research on the relationship between nutrition and gene expression in the development of hypertension and chronic kidney disease. For example, something that we have not talked about here, we identified the gene PRR2, and we observed changes in the circadian rhythm of that gene with respect to micronutrient intake. Therefore, one extension or follow-up to the study could be the investigation of the effect of meal time on blood pressure. It will also be interesting to determine if food consumption late at night or throughout the night contributes to the phenomena of non-drooping blood pressure and or nocturnal hypertension, and whether this can be linked to the signals we report in this study. That line of investigation could be extremely important. I was reading just recently, I think it's yesterday, saying that blood pressure increase at night. Individuals who tend to have blood pressure that is roughly normal on day but then have increased blood pressure at night are at a double risk of having a heart attack. So that's what I wanted to convey to you today. I hope I got the message across that there are transcriptome profiles, meaning gene expression profiles that are linked to sodium intakes, or in more general terms to poor diet. And we have established a relationship between the transcriptome profile related to poor diet. We link it to hypertension and systolic blood pressure and kidney function in this analysis. I would like to acknowledge the contribution of some of my colleagues. First of all, I want to convey my acknowledgements to Dr. Gaye Gibbons, who is the PI of the studies that I presented here and who trusted me with these analyses. And I want to thank also Gabriel Goodney, a bioinformatician who works with me, Dr. Malak Abbas, a molecular biologist, Dr. Polly Joseph, who I collaborate with, our clinical team, Dr. Jose Vargas, Commander Nicole Plas, and Michelle Afrier, and all the trainees who worked with me on this. I have had trainees that came work with me and left. They all contributed to this work. And I would like to thank some of my mentors at the NIH, Dr. Sharon Davis and Charles Martin. Thank you.